Hello. Yeah, everyone. I'm Chris Daldorf. I'm a student in Dr. Paulson's lab at UCSD. I'm presenting today on this paper, Adaptive Evolution of a Minimal Organism with a Synthetic Genome. All the authors are listed there. So, without further ado, I'll just jump into it, essentially. So sort of the overall goal of this paper was to see the feasibility of evolving these synthetic organisms through adaptive laboratory evolution. If you're not familiar, that's essentially just throwing a strain on some medium, uh, letting it evolve for a bit, changing the medium, letting it evolve, and sort of repeating that process for a while. So we ran that for about 500 generations in 10 independent replicates. And sort of got expression data and sequence data to sort of see what are these evolutions doing, what are their targeted goals. So sort of overall results, this evolution, as you can see in panel B here, resulted in these large and small colonies, which there really are no clear differences to, but I just think is interesting to note. But sort of the good results, evolution is clearly able to significantly improve growth rates. They improve similarly in both colony sizes, and they sort of start to approach that of syn 1.0, which is just sort of really promising to show that evolution can improve growth rates even in this minimal organism. I'll talk a little bit about mutation results now. So sort of a big concern for this paper was trying to analyze using AL. AL is really dependent on using independent lineages to look for convergent mutations to imply causality of those mutations. But if the strains are hypermutators, it's really difficult to separate what are just hitchhikers from what are actual causal mutations. Very surprisingly, syn3A was not a hypermutator in this evolution. There's an average of 8 mutations per strain with a maximum of 10, despite most of the DNA repair genes being gone. So it's really great for the sort of future promise of applying AL to syn3A. Mutations show this clear preference from C to G to A to T also, which is what that left half of panel C there on the top right is showing, which makes sense. Syn3A already has this heavy preference for AT. It's cheaper to replicate. So sort of anything evolution get away with to get cheaper, it'll do. RPOC mutations are also found across nearly all strains, which I'll talk some about earlier, but I wanted to point out now. Sort of other general results from where these mutations are is transporters are a really common target, less so translation and some of these other sort of pie chart type results you can see in panel D. Indels were somewhat common, and they all reduced genome size. There's only one addition of a base pair mutation observed. So it's interesting that even after all this sort of synthetic reduction in genome size, evolution will naturally even push that a little further. So sort of next up would be, what are these specific mutations? TET-M was a common target, which is a antibiotic response gene. There were a lot of upstream mutations of it, which reduced its expression, as I'll talk about shortly on the next slide. Uncharacterized transporter mutations showed a lot of convergence. One of those is shown in panel A, with a lot of these mutations highlighted in red on these transmembrane subunits. So it's sort of interesting to see even these uncharacterized transporters are clearly having some directed effect that hopefully future studies can better explain. FAKA mutations were also common. It's a fatty acid kinase. It was mutated in 4 of the 10 endpoint strains. It's a little impossible to say what these mutations are doing, but again, just sort of good to see convergent mutations in ALs. Lipoprotein mutations were also seen. That's what panel B is displaying. This long TA repeat upstream of one of their start sites was found to be reduced in a handful of the evolutions. It slightly increases expression of those, so presumably it's having something to do with ribosomal binding, or I guess RNAP binding, but there's also a lot of convergent mutations or deleterious mutations to a lot of unknown function genes, which is sort of interesting to show. Even these genes that we don't know what they're doing other than improve growth rate given they're in syn3A, have some clear function that either syn3A is trying to change or get rid of at this point through evolution. So we have expression profiles on, I think, most of the strains. You can see there about 1/3 down or 2/3 down in that dark blue. TET-M is really downwardly expressed in AL5, which is the one with the most of those upstream mutations. I want to point out one thing, as you can see sort of two clear clusters in the strains here. AL1 through 3 and 4 through 8 really sort of gather with each other. We think that has something to do with the phase in which they were collected. As you can see, that wild-type stationary phase sample in there really clusters closely with 1 through 3, while it does not with 4 through 8. Another sort of interesting note is that sort of large and small colony samples there really are very similar to each other. The downregulated genes largely consist of stress-related genes and DNA repair type genes, while the upregulated genes really consist of ribosomal subunits. There's not enough data to run ICA, which is sort of a big Paulson lab thing, but what we can see here is sort of this downregulation of stress-related genes and upregulation of ribosomal subunits. So the reason I mentioned that and the RNAP mutations is this wasn't exactly part of the paper, but was sort of just sort of interesting in my own conclusions from it. RNAP mutations are really common across laboratory evolutions. You can see that on the left, the most common genes mutated, and RPOB falls as number 4, and RPSC shortly follows it. On the right here is an image of RNA polymerase and where these mutations lie. The white to red color are allelome differences in the wild type, and the blue color are LDB mutations, which are clearly showing some clustering to a few regions towards the center of RNAP, most particularly towards PPG-PP binding site. These mutations have been closely associated with those changes I mentioned earlier of upregulating growth-related genes and downregulating stress through imodulons, which, if you aren't familiar with, essentially just think co-regulated regulons of genes or just groups of genes. This figure is from sort of a manuscript in process about this, but essentially, this trade-off is found in a wide variety of species. On the right there, you can see 8 of them and where sort of it lies. The x-axis in all of these are the fear-related genes, and the y-axis are all the greed-related genes. And essentially, as you move up and to the left in all these curves, growth rates go up. Panels B and C show sort of how these imodulons, or just think groups of genes, sort of conserve between each other, and these greed-related ones are essentially just all ribosomal subunits and are really found in all species everywhere, and the fear ones less so, presumably due to varying natural stresses found in their own environments. But the point I bring this all up to make is that it's very fascinating to me, at least, that syn3A, even despite all of its sort of unnatural changes, so to say, still has this trade-off in it. Perhaps with more data, it'll be more clear for syn3A, and perhaps you could run ICA on it. But for now, it's sort of just promising to show it is still meeting these sort of basic bacterial trade-offs that exist. So sort of just general conclusions and potential follow-ups. The big one is that the evolution of this minimal cell is feasible. It does all the things you want to see in ALs. It improves growth rates. It shows convergent mutations. It doesn't hypermutate. It was very good for identifying these rate-limiting genes that would be great targets of future studies, sort of showing even these unknown functions are clearly having some specific effect. And then also it just shares these transcriptional patterns to sort of non-synthetic bacterial strains, showing that sort of the mechanisms in these minimal genomes are still similar to those of more complex, showing that they're really conserved even with such a reduced full genome. As far as future directions, I think sort of better modeling by AL would really be helped by a better formal definition of growth medium. Obviously, it's kind of hard to grow this minimal cell. It isn't, it's heterotrophic for so much, but and also different sort of follow-up would be varying these ale conditions. Stress ales are really common, gene knockout ales, longer evolutions. There's clearly a lot more to figure out here if sort of conditions were varied more. But yeah, that's really it. I put a link to the paper there and a bit.ly link if you're curious, or you can sort of Google the paper name from the start. But yeah, I know I probably talked too fast and ended early, but I always end up doing that.
Chris, nice. So, as many of you may know, there were two ale papers that published almost on top of each other. I'm an author on both. So there was the one that Chris just talked about, done at UCSD, and then at Indiana University, Jay Lennon's group did another one. And there were some. Chris, could you talk about the differences in the two papers?
I have not spent much time with the other one, so maybe you would be better to talk about it. I know they found pretty similar results, which was exciting, but I can't remember much more than that.
The big difference is that FTSZ mutated in their experiments and not in yours. And in the wild type cell, the cell got larger with FTSZ mutations. And in the minimal cell, the cell got a little smaller with FTSZ mutations. But the protocol for sampling was different. So in the case of Indiana, they didn't agitate their cells and they isolated the cells that they were moving to a new tube every other day or so from the bottom of the tube, whereas your cells were constantly agitated and probably you were taking planktonic cells.
Huh.
But the differences in the outcome is kind of striking, and it probably deserves some thinking about, but clearly the difference in sampling gave radically different results. You also, so I can see that for a pathogen, no one ever tried to serially pass Mycoplasma mycoides, the cell on which the minimal cell was based, in order to optimize it for growth in laboratory media. People wanted to keep it as close as possible to the pathogen from which it was derived, because that's what most people were studying. Is that the case for most of these other organisms that you were looking at?
Yeah, so most of my data is all from sort of laboratory ale conditions, which, yeah, definitely shows some differences. I know that was a point in this paper of why there's probably so many transporter mutations, is adapting to sort of laboratory conditions from its natural environment. So yeah, I mean, this trade-off is definitely way more present in laboratory, where at least it's well more controlled, at least. But yes, essentially.
But you would think, for instance, that the coli example that you gave, that likely the coli you had been passaged a great deal since its original isolation, however long ago, and probably the same for many of those other organisms that you were working with.
Yeah, so there's a variety on that list, and definitely for E. coli, it's been spent so long in laboratories at this point that it's so efficient on M9 compared to most other species. And some of these are also similar to that. Less would be like Sophilobus down there really isn't. But there's also an Archaea species, or maybe that is, I'm forgetting at this point, but that showed a weaker version of the trade-off. So yes, essentially, I mean, this is definitely more clear in laboratory settings, but I mean, that's where all the data is, so kind of unavoidable to study it in that way at scale.